So anyway, I have anyway it is advancements in advances in phytoremediation. Anyway, we will discuss the latest trends in the area of phytoremediation. So this is the content of the presentation. We will start with the definition of phytoremediation and also we'll discuss the applications. Then the session is will be divided into two different topics, subtopics. One is phytoremediation of soils contaminated with organic chemicals, and the third subtopic is phytoremediation of soils contaminated with heavy metals. Because you may be knowing that our soil, especially in industrial areas, in other areas, are contaminated with the different types of organic substances, organic chemicals, which are some of which may be toxic in nature, and also soil in many parts of the world are contaminated with the heavy metals. For example, heavy metals like cadmium, mercury, lead, nickel, zinc, they all belong to the category of heavy metals, and heavy metals are also toxic in nature. So there is a need to remediate these soils. So first we will see the definition of phytoremediation. So as I told you earlier, as the name indicates, it's the use of vegetation. It's the use of plants for in situ treatment of, it can be applied to contaminated soils, sediments as well as water. So the use of vegetation for the in situ treatment of contaminated soils, water, etc. is known as phytoremediation. And this technique can be applied to sites containing, one is organic substances. Organic substances, for example, different types of organic residues are found in our soil. And we talk about organic substances which are not biodegradable in nature. The substances which are biodegradable will easily get degraded over a period of time. But the organic substances, some of them are referred to as persistent organic chemicals, persistent organic pollutants. They are resistant to biodegradation. Such organic substances will remain in soil for a very long time, and also it will get leached to groundwater. For example, a best example is DDT. I think most of you have heard of this, what we call, we can call it an insecticide. It is still widely used in our country for mosquito control. This DDT, diphenyl trichloroethylene, DDT is there. DDT it is an organic substance. Such substances, okay, some of them they can be referred to as recalcitrant substances or refractory organic. Such names are also used. Then another contaminant is the nutrients. Nutrients, we know that we apply fertilizer to soil to improve the nutrient level of the soil, so that these nutrients will be available for the plants. Nutrients in the form of nitrogen, nutrient in the form of phosphorus, they are all added to the soil. And anyway, by natural mechanisms also, some of the nutrients are added to the soil. But these nutrients, the excess nutrients present, they remain as a contaminant in the soil. Of course, it will get leached to groundwater also. Then the third category is the metal pollutants. Earlier I mentioned about the heavy metals. All the heavy metals, they are, the heavy metals are of what we call anthropogenic origin. Many of them result from industrial activities, metal pollutants. All these heavy metals like mercury, cadmium, then lead, nickel, zinc. All these are called heavy metal pollutants. So whatever component, only thing is these metals should be accessible to the roots of the plants. Okay. Anyway, that when we go for a vegetation, the plants, that roots of the plants have a unique ability to absorb or sometimes absorb these materials from soil. So, and also they access these materials, these contaminants found in soil, then they sequester them, some kind of immobilization, some kind of it is not immobilization, it is some kind of it may be some kind of degradation or some kind of transformation. All those processes can take place. Sequestration, then it may be degraded, immobilized, or even metabolized in the system. It may, these substances, the plants may detoxify these materials and use them as a source of food. It will get metabolized in the plant system. So that's the meaning of phytoremediation. And during the last few years, a lot of work has been done, especially at the laboratory level. And now we have a greater understanding of the uptake and metabolism of this organic xenobiotic chemicals. Xenobiotic chemicals, it's another name used for the substances which are not easily biodegradable. So during the last 30, 40 years, a lot of work has been done regarding the uptake and metabolism of these xenobiotic chemicals by plants. And some of the examples are chlorinated solvents, pesticides, explosive compounds, and all that. You have heard of, some of you might have heard of TNT, which is trinitrotoluene. So it is an explosive compound, and at the same time, it is a xenobiotic, not easily biodegradable substance. So these, and also these chemicals contaminate a large number of hazardous waste sites. Anyway, during the second half of 20th century, and also even during the first half of 20th century, lot of industrial development has taken place in the West. And also after independence, countries like India also went for large-scale industrialization. Large number of manufacturing industries, especially in the chemical and petrochemical manufacturing sector, were installed. And all these industries generate hazardous waste containing these organic substances. So, in fact, there is no, we cannot say a satisfactory method, a foolproof method for treating these hazardous waste. So nowadays we go for safe landfills and all that in the case of hazardous waste, just like municipal solid waste. But anyway, in a safe landfill, anyway, that leaching will be prevented due to a great extent. There won't be, anyway, groundwater and soil will not get affected in an engineered safe landfill. But in earlier days, that was not the case. These substances used to be dumped on open grounds. Even in the West, that was the practice, and the large areas got contaminated. So now there is a need to reclaim those areas, and techniques like phytoremediation becomes relevant in such a context. Then why it is so popular? There are three reasons. One is cost effectiveness, because you can go for vegetation. It is not a costly affair. Planting trees is not an expensive thing. So cost effectiveness. Then it has aesthetic advantages. You can see Anyway, suppose it is a hazardous waste dumping site. There you are in fact growing trees there. So that area will have an aesthetic advantage. When trees come up in the area, that area will have a good look. So that is another advantage. And also these techniques have long-term applicability. That is also there. So these are the reasons. These are the reasons for which make phytoremediation very popular. Now, applications. Where can we apply these phytoremediation techniques? One we have already seen in hazardous waste sites, where other methods of treatment are too expensive or impractical. In such waste sites, you can make use of these techniques. Then the second category is low-level contaminated sites. They are low level. Anyway, the contamination is not at a very high level. So there we can go for some kind of a polishing treatment over a long period of time. So this polishing treatment can be brought about by phytoremediation, by these vegetation that grows on these low-level contaminated sites can bring about this polishing treatment. Then, of course, we can use phytoremediation in conjunction with other technologies. That is also possible. Anyway, of course, you can think of other remediation technologies. Along with that, you can try phytoremediation also. So these are the areas where we apply phytoremediation techniques. Now, anyway, it has its own advantages, but at the same time, the phytoremediation technology has limitations also. What are the limitations? We should be aware of the limitations. It's not a panacea for all the problems. You cannot solve all contamination problems using phytoremediation. It has its own limitations. So one major limitation is the potential for introducing the contaminant or its metabolites into the food chain. That is the biggest problem. Anyway, the plants absorb the contaminant. This contaminant substance, it may be a hazardous chemical or a heavy metal. Then in the plant system, it may undergo metabolism and it will result in the generation of metabolites. These metabolites also may be toxic in nature. So there is a possibility that the contaminant or its metabolites are introduced into the food chain, because anyway, you know the principle of the food chain in the environment. So in the food chain, naturally, plants are primary producers. So these herbivores and all that, they eat these plants, and as we move up in the food chain, when these animals are eaten by other animals, they move up in the food chain and it may ultimately reach the human beings. So that danger is there. Then the second problem is the long cleanup times. Long cleanup times required to achieve regulatory action levels, because regulatory levels are very stringent. Suppose anyway, in our country we don't have a proper legal framework for decontamination of hazardous waste dumping sites, but in the West these regulatory requirements are very stringent. So when you clean up a site, you will be given a target. Suppose it is a heavy metal contaminated site, and suppose you are using phytoremediation technique. So maybe the legal requirement may be over a period of time of 1 or 2 years, the level of cadmium, the concentration of cadmium in soil should be brought down by say 30%, 40%. Like that, they may give fixed targets. So it may be difficult to achieve these targets because the cleanup times are much, they are pretty long. So that problem is there. Then toxicity encountered in establishing and maintaining vegetation at waste sites. That is also there because these organic chemicals as well as heavy metals, many of them are toxic in nature. So the plant should be able to tolerate that toxicity. Then only we can establish and maintain at the waste site. So we have to select the vegetation very carefully. Now let's see the role of plants. So plants, different types of plants we use. For example, a very popular plant species that we use in soil remediation is it is widely available in our locality also. It is vetiver grass. Some of you might have carried out studies using vetiver grass. Ramacham എന്ന് പറയുന്നതാണ് vetiver grass. Vetiver grass is anyway, lot of studies have been carried out to establish the potential of this vetiver grass as a phytoremediation agent. So these plants, plants of such nature, they have shown the capacity to withstand relatively high concentrations of organic xenobiotic chemicals without toxic effects. So the plant should be able to tolerate that toxicity, so they can withstand. Many plants have shown that capacity. Even this vetiver grass has that capacity. Then some cases, they will take up and convert these chemicals to less toxic metabolites. So as part of their metabolic process, the plants convert these chemicals into less toxic ones. Then of course they stimulate the degradation of organic chemicals in the rhizosphere. Rhizosphere refers to the root area, that root area of the plant. So these plants stimulate the degradation of these substances in the rhizosphere by the release of root exudates and enzymes. Anyway, these roots also release certain enzymes and other substances. They're called exudates and enzymes. And the resulting buildup of organic carbon in the soil. Okay, so organic carbon will build up in soil in that process. But in the case of metal contaminants, plants show the potential for what we call phyto-extraction. We will have a detailed discussion about phyto-extraction later when we discuss about metal contaminants. So for plant metal contaminants also, they have a good, they have the potential for phyto-extraction, and also they are found to filter metals from water onto root systems. That is also happening. Now in this table, you can see a list of typical plants used in various phytoremediation applications. Anyway, these application techniques we will discuss in later. Detail later. Phytotransformation, rhizosphere bioremediation, phytostabilization, phytoextraction. See, phytoextraction is applicable for metals. So the media is soil, sediments, and brown fields. Metals like lead, cadmium, zinc, nickel, copper, they can be removed from soil and sediments using phytoextraction. And the plants, the popular plants are Indian mustard, sunflowers and all that. They are found to be very effective. Then rhizofiltration. Rhizofiltration is for groundwater and wastewater through constructed wetlands. In constructed wetlands, here you can go for this high water hyacinth and all that. Okay, and also emerging aquatic plants like bulrush, cattail, coontail, pondweed, and all that. Okay, even arrowroot is a good As far as organic chemicals are concerned, we have techniques like phytotransformation and rhizosphere bioremediation. So these are the contaminants usually. Contaminants that can be removed from soil or groundwater, herbicides. Anyway, it is applicable even for landfill leachate. In engineered landfill systems, we collect the landfill leachate. No? That landfill leachate may contain all these substances, so we can treat this landfill leachate using these technologies. So the contaminants found are herbicides, chlorinated aliphatics. The example is trichloroethane. Then aromatics like benzene, toluene, ethylene, xylene, and all that. Then ammunition waste like TNT, RDX, all those waste. Nutrients like nitrate, ammonium phosphate. Then here you can go for these phreatophytic trees belonging to the salix family, including poplar, willow, cottonwood, grasses, Bermuda grass, and all that. So these are the typical plants used for various bioremediation applications. Now we will discuss in detail about phytoremediation of soils contaminated with organic chemicals. So the first aspect that we are going to discuss is the sorption of organic chemicals to roots. Sorption is, sorption belongs to, it is mostly adsorption. Adsorption of these substances to roots. Of course, absorption can also occur. Absorption is a mass transfer operation, and adsorption is also a mass transfer operation. And adsorption is a surface phenomenon. Okay, we have physical adsorption called physisorption and also chemisorption. So these organic chemicals may soak to roots, and then the chemicals that are absorbed by or soaked by the roots can be taken up, translocated, metabolized, or transpired by plants. That's what's happening. They take up the substance, translocate it to other parts of the system, then metabolized or transpired. Transpired is volatilized by plants. Then the first step is sorption to plants. So what will happen? Suppose chemical contaminants in soil come into contact with the roots. Then definitely they may sorb or bind to the root structure and cell walls. Anyway, these are all living organisms. Plants are living organisms made of cells. So the chemical contaminants may sorb or bind to the root structure and cell walls. And hemicellulose in the cell wall and lipid bilayer of plant membranes can bind hydrophobic organic chemicals effectively. These are the results of some studies. And also the okay. So hemicellulose, then again, you know that plants contain cellulosic material. And also we have a constituent called hemicellulose. Hemicellulose is found in the cell wall. We know that all living organisms are made of cells, and these cells have a cell wall. So hemicellulose in the cell wall of the plant, and also there is a lipid bilayer in the plant membrane. So both of them can bind hydrophobic organic chemicals effectively. So they will not go away with water. So these hydrophobic organic chemicals can be bound effectively in the cell wall and the lipid bilayer of plant membranes. but such sorption should be relatively reversible. It should be a reversible process and can be measured using standard sorption isotherms. So here we are talking about adsorption, adsorption isotherms. You might have heard of adsorption isotherms. So these sorption on roots can also be modeled using the standard adsorption isotherms. So this is it's an isotherm. It's a sorption isotherm after 48 hours for 1,4-dichlorobenzene in hydroponic solution with the fresh hybrid poplar roots. Poplar plants are used for the purpose. But anyway, this here the dichlorobenzene was used for the study, and this isotherm pertains to that. Here it has a very good R square value and all that. It's found to be a good candidate isotherm for the sorption of dichlorobenzene on hybrid poplar roots. Then, anyway, we'll continue the discussion on organic chemicals and sorption. As we have seen earlier, during the last 30-40 years, a lot of experimental work has been done, not at the field level. Most of the studies are at the laboratory level. So, a group of scientists, Briggs et al., defined the root concentration factor. It is called RCF, root concentration factor, as the ratio of organic chemical sorbed on the root to that in hydroponic solution. So all the material may not come to the root. The ratio of the organic chemical sorbed on the root to that in the hydroponic solution. Hydroponics is a unique way of cultivating plants. It's in cultivation in plants, in fact. So the adsorption isotherms are used there. Linear isotherms are used there. These slopes are measures of the RCF. RCS is the root concentration factor, and they have the units of liters per kilogram. That way we express this root concentration factor. Then we have seen hydrophobicity. Hydrophobic substances can easily be fixed on other materials, so it can be easily fixed by adsorption. So hydrophobicity was related to the octanol water partition coefficient. And water partition coefficient is there. Octanol water partition coefficient of the true organics. And also, and again, these are some RCF values. These root concentration factor values are shown here. Liters per kg. Last column is this RCF. So some of the substances have a very high RCF values. For example, aniline, then pentachlorophenol and all that. Then these hydrophobic chemicals, we are talking about the hydrophobic chemicals first. They are water repelling chemicals. They don't have, there is no love lost between this this content. Okay, so these hydrophobic chemicals, they have a tendency to partition into organic phases. So that's not the only mechanism. So they will partition it into organic phases. So other mechanisms are also at play in binding the chemicals to roots. So the other mechanisms are like specific sorption, not chemical species, and enzymatic transformation by membrane bound protein. So the specific sorption at chemical sites and enzymatic transformation by membrane bound proteins are other mechanisms of potential importance. These mechanisms, so we have specific sorption at chemical sites and enzymatic transformation, that is a part of metabolism, and enzymatic metabolites are formed, and they are carried out by membrane bound proteins. So you have seen that table. Some of the chemicals are they are largely highly soaked to plant tissues. If the RCF value is more than 10 liters per kg, then it is highly absorbed into the plant. So here you see. Okay, so some of these chemicals have very high RCF value, like aniline, then pentachlorophenol, even dichlorobenzene. They all have high RCF values. So because of their these high RCF values of high RCF value is because of hydrophobicity. But aniline and phenol, they bind tightly to roots because of specific sorption and enzymatic transformation. So we can easily remove these materials by phytoremediation. Then we know that some of the contaminants found in soil, uh, they get absorbed by the plant. The roots take up a plant, and they're transformed rapidly at the root surface by extracellular enzymes or by membrane bound process. That's what is happening. Okay. Okay, then organic chemicals. Then comes uptake and translocation of organic chemicals. Anyway, the plants, the roots absorb or adsorb these substances, these organic chemicals from soil. The next step is uptake and translocation of these chemicals. So these rooted vascular plants, they must take up water and nutrients for growth. Plants will grow only if nutrients and water are available. Then nutrients are transported into cells through so this now coming to uptake and translocation. So we have different types of rooted vascular plants. So they must take up water and nutrients for growth, and these nutrients are transported into the cells through channels in membranes or through membrane bound proteins. We have a group of proteins called membrane bound proteins. So through these channels, the nutrients are transported into the cells. Okay. Then these base metal cations, they are taken up by active transport mechanisms, whereas organic chemicals may be taken up by plants via passive uptake through the cell walls and membranes. So the uptake through cell walls and membranes is referred to as passive uptake. Okay, it's usually diffusion is a mechanism, and in this case, there may exist an optimum hydrophobicity that allows the chemical to bind to the lipid bilayer of the membrane, because it is hydrophobicity. It is a water repellent substances. So it is, but it will get bind to the lipid bilayer of the membrane. But it may not be a very strong link, but still there will be a link. Then this direct uptake of organics by plants, it is a surprisingly efficient removal mechanism. So it happens in shallow contaminated sites. So the plants grown on shallow contaminated sites, they directly take up, or they bring about a direct uptake of organics by the plants. With anyway, in the case of shallow contaminated sites with the moderately hydrophobic chemicals. So a criterion for an index of hydrophobicity is this log KOW, log KOW, when it is in the range of 1.1 to this 1 to 1.35, then it is direct uptake is possible. So the organics which can undergo this direct uptake are benzene, toluene, ethyl benzene, and xylene. They together they are called BTEX: benzene, toluene, ethyl benzene, and xylene. Then chlorinated solvents, short chain aliphatic compounds, they all belong to this category. So these hydrophobic chemicals, they're hydrophobic chemicals with this low KOW. It is we can call it a separation constant. So the separation between two phases, that organic substance and water. So log KW is greater than 3.5. They are strong hydrophobic chemicals. So they are bound so strongly to the surface of roots and soils that they cannot be translocated easily within the plant. Then there are substances that are quite water soluble. There the low KOW value will be less than 1. And these are not sufficiently soaked roots nor actively transported through plant membranes. Then these hydrophobic chemicals with this low KOW value greater than 3.5, they are candidates for phytostabilization and rhizosphere bioremediation by virtue of their long residence time in the root zone. So they can be, they can undergo the these hydrophobic chemicals can undergo the phytostabilization process or rhizosphere bioremediation. And there are some empirical relationship. So this uptake of chemicals into plants through roots, this depends on 3 factors. One is the plant's uptake efficiency. Here it is represented as TSCF, efficiency of uptake. TSCF, it is transpiration stream concentration factor, TSCF, transpiration stream concentration factor. It's a dimensionless quantity. T is the respiration rate. TSCF into T. T is the respiration rate in liters per day. And C is the soil water concentration of chemical in milligram per liter. So this way we can calculate the rate of chemical uptake by plant. Then this TSCF, the transpiration stream concentration factor, so it is in fact the uptake efficiency for rooted vascular plants. That is known as transpiration stream concentration factor. And TSCF, if we consider it mathematically, it is the ratio of the concentration in the transpiration stream of the plant to the concentration in soil water. So this over a period of the concentration, this concentration may come down. Okay, so TSCF. And this TSCF, it is the ratio of the concentration in the transpiration stream of the plant to the concentration in soil plants. Because these plants constantly transpire, because they have to send out this water vapor and carbon dioxide and other substances. And this the value of TSCF depends on physicochemical properties of the plant, chemical speciation, and the plant itself. And some of the measured values are given here. Anyway, that table is known. Anyway, typical values are available in literature. And this TSCF can vary from 0 to 1. 0 is no uptake, and 1 is uptake at the same time transportation as the soil water concentration.
Excuse me, sir.
Yeah.
May I ask one doubt here?
Yeah.
Sir, in what condition TSCF will be 0? That means plant is not making any uptake, or
there is no uptake. If it is 0, there is no uptake. Yeah.
Okay. So that plant is not suitable, or in what way we will be inferring that result? 0 uptake means?
We can conclude that plant is not suitable for that particular condition. Yeah.
Oh, thank you, sir.
So it is again more about this TSCF. So it has relationship with transpiration rate. So transpiration rate is a key variable that determines the rate of chemical uptake for a given phytoremediation application. And it depends on various factors. The factors are plant type, leaf area, nutrients, soil moisture, temperature, wind condition, and also the relative humidity. These conditions, these parameters, these factors influence the transpiration rate. And high transpiration corresponds to rapid uptake. So this is the reason why fast growing plants, they are popularly known as phreatophytes. These plants are not very popular in our country. Hybrid poplars, willow plants and all that. They are frequently employed in phytoremediation applications in the West. Then, now we have to discuss. So we have seen sorption of the organic substances, then uptake and translocation, and now we will talk about enzymatic transformation of organic contaminants. So the organic substance that is taken up by the plant will undergo enzymatic transformation. So it usually call it phytotransformation. It refers to the uptake plus the subsequent metabolism or transformation that take place in the plant. That is called phytotransformation. So there are 3 phases in this transformation. Once an organic chemical is taken up and translocated, it will undergo 3 phases. 3 phases of transformation. One is conversion, then there is conjugation, phase 2 is conjugation, and the third phase is compartmentation. In conversion, mostly chemical reactions take place, oxidation, reduction, hydrolysis reactions can take place. Then conjugation, the substances that result from the conversion mechanism, the conversion phase will join together with substances like glutathione, sugars and amino plants found in plant cells. That is conjugation. Then compartmentation. Then here the conjugates from phase 2, they are converted to other conjugates and also deposited in plant vacuoles or bound to cell wall and lignin. So this vacuole and all, they are all part of the plant cell structure. Then this enzymatic transformation, sometimes these phase 3 conjugates are referred to as bound residues because of their inability to be extracted by chemical method. So they may, it is a sort of immobilization. They get bound to the bound to the cell, and these conjugates are likely, most likely they will be covalently bound to stable tissues in the plant. So different mechanisms have been, a lot of studies have been, mostly as I told you, these are all studies are carried out in the laboratory. This is chlorinated aliphatic compounds are found in hazardous waste dumping sites. But another, a very popular example is trichloroethylene. So they have been, so these substances, by enzymatic transformation, they may be converted to substances like gases like carbon dioxide and less toxic aerobic metabolites. So simpler metabolites will be formed. Like from trichloroethylene, you get a trichloroethanol, trichloroacetic acid, dichloroacetic acid, and all that. Okay. And these products are consistent with those found in human liver. So there is a remarkable resemblance between the metabolic process taking place in plants and animals as far as these substances are concerned. Even we know human liver is a detoxification mechanism in the body. Liver can detoxify many substances. Anyway, it has a certain carrying capacity. So TCE even human liver has the capability to destroy TC by cytochrome, this enzyme P450. So the same enzyme is found in plants as well as in enzymes. So because of this reason, plants are sometimes viewed as green livers in terms of their enzyme biochemistry, because of that remarkable resemblance with the human liver. Then other enzymes, for example, anyway, you know that in living systems, large number of enzymes are found. Even in the human body, we all survive due to the action of enzymes. So enzymes like nitroreductase, lactase enzymes in plants, they can break down even TNT, 2,4,6-trinitrotoluene. It's an ammunition-based, explosive-based, and may incorporate the broken ring structures into new plant material or organic detritus that become a part of the soil organic matter. And and also there are detoxification mechanisms in plants, and these mechanisms may transform the parent chemical to non-phytotoxic metabolites stored in plant tissues. That is also happening. So there is a need to have a thorough understanding of pathways and end products of enzymatic processes. A lot of biochemistry is involved here, enzyme kinetics and biochemistry is involved. And a thorough understanding will simplify toxicity investigations of in situ phytoremediation. Then enzymology, biochemistry, chlorinated aliphatic degradation. Anyway, the comparison, that resemblance between the enzymatic processes taking place between between plants and animals is mentioned here again. Then, for example, this dechlorination pathways. Earlier we talked about this trichloroethylene. So the major dechlorination pathway for chlorinated ethylenes, they are they are found in waste dumping sites in the West. That's why a lot of studies have been reported. So the major dechlorination pathway for chlorinated ethylenes involves the formation of what we call epoxides with polychlorinated ethenes such as TCE and PCE, alternatively being conjugated to glutathione. In the first phase, that dechlorination activity will take place. In the second phase, they will form a conjugate with glutathione, sugars, and all that. So here this enzyme P50 is involved in epoxide formation, whereas another enzyme called glutathione S-transferase, GST, it catalyzes reactions with glutathione. Okay. Then this TC is in fact one of the more studied compounds, and metabolites commonly reported in experiments with rodents. Even experimental animals have been used in the studies, and experiments with the rodents showed that the metabolites are chloral hydrate, trichloroethanol, dichloroacetic acid, and trichloroacetic acid. And this is a transformation TCE metabolism pathway in mammalian systems. So all those substances are formed. So this is that TCE getting that all the phases are shown here, and different types of compounds are formed, and even aliphatic compounds are formed. They also result from this metabolism. It's a pathway. Anyway, just for information. So it is more about the phytotransformation. Then in the case of pesticides, we have already seen the transformation of the TCE, compound called TCE. Now pesticides, some studies have been carried out on pesticides in crop plants. These compounds also undergo a series of metabolic processes. And they're also the first phase. Anyway, we have three phases. In the first phase, functional groups like hydroxyl group, amino group, then this mercaptan group, they are introduced, and the first phase can occur by oxidation, reduction, or hydrolysis. Then in the case of highly lipophilic compounds, oxygenation is a typical reaction of the first phase. These fatty compounds and all that, fatty acids, lipophilic compounds. And anyway, the typical reaction after the first phase, the solubility of the compound will increase. Then second phase involves conjugation with that we have already seen, D-glucose, glutathione, amino acids, resulting in soluble polar compounds. Then third phase of plant metabolism is compartmentation. So first phase, that degradation. Then the second phase is that binding together with these conjugates. And the third phase is compartmentation or storage. So there is a difference. Mammals can excrete unwanted compounds, but plants do not have that option. So soluble metabolites are stored in the vacuole or as part of the cell wall materials. So unwanted compounds cannot be sent out. So they are also stored in the vacuole of the cell, or they may be stored as a part of the cell wall material. And the transport of glutathione conjugates into the vacuole has been demonstrated in barley cell cultures in some studies. Okay. And there are several potential mechanisms for the uptake and transformation of chlorinated aliphatics in a plant soil system. Several potential mechanisms are there. And possible mechanisms include microbial transformation. Even microbes can also act on it. In the root systems, in the rhizosphere, these compounds will be made more micro-friendly. They will be transformed into substances which are friendly to the microbes. Then uptake of the chemical, or its metabolites into the root, xylem transfer of the compound to the leaves, and it will move up to the leaves. Then volatilization from the leaves, then foliar uptake of chemicals from air, phloem transfer, mound residue formation. These are all possible mechanisms. Several mechanisms, anyway, we don't know what is actually happening, but scientists have come out with possible mechanisms. And all these mechanisms may prove important in phytoremediation of sites contaminated with this chlorinated aliphatics. So this is a potential uptake and transformation pathway of PCE, trichloroethylene, in a plant soil phytoremediation system. Here you can see the roots, the stem, and the leaves. Okay, different mechanisms take place. Soil absorption, then here in the stem, the transport is taking place. Then it will reach the xylem transport. It's a two-way mechanism, phloem transport. Ultimately, from the leaves, we can have volatilization, then in-plant transformation and bound TC residue can be found in the leaves. All those mechanisms can happen. Now there are attempts made to improve or develop genetically modified plants that are able to, that have better efficiency, take-up efficiency and degradation efficiency. And you know that these transgenic plants are created using genetic engineering, what we call genetic engineering. So, and some of these genetically engineered plants are found to be more efficient than the ordinary plants for remediating soils with xenobiotic chemicals, these pesticides, chlorinated aliphatics, aromatics, and all that. Okay, so transgenic application of transgenic, several transgenic species are being developed with phytoremediation applications in mind, and these plants are said to have higher tolerance to many of these organic substances. These are the. Then another issue is the toxicity issue. We know that all these chemicals are the organic chemicals found in soil are toxic in nature. And also now the toxicologists, plant scientists, they are focusing on the relationship between plant transformation of xenobiotics and phytotoxicity, but this relationship is not yet completely understood. And some studies in mammalian systems, in animal systems, the activation of TC through the epoxide intermediate is found to produce carcinogenicity. Some kind of cancer-causing compounds are generated through this mechanism. But in plants, it's not, this relationship is not fully understood. Okay, so that's the second part. So we have discussed in detail about phytoremediation of soils contaminated with organic chemicals. Now the third part of this lecture is phytoremediation of soils contaminated with heavy metals. That is another problem. Most of these dumping sites are contaminated with heavy metals. So heavy metal, the presence of heavy metal, it's a big problem. Abundance of heavy metals in the environment has increased tremendously during the past decades, and it's really an area of concern for environmentalists as well as health specialists. And anyway, we have already seen the common heavy metals include cadmium, mercury, lead, arsenic, zinc, copper, nickel, and chromium. And these metals or metalloids, they can originate from either natural or anthropogenic sources. There are various sources, anthropogenic sources, like for example, oil and gas industries. Oil and gas industries generate residues containing these heavy metals. Then use of phosphate fertilizers in agriculture. Phosphate fertilizers are manufactured using rock phosphate. Rock phosphate is the major raw material, and rock phosphate contains, it is a naturally occurring material. So along with the rock phosphates, compounds like these heavy metals like cadmium, mercury, zinc, nickel, etc. may be present, and they may end up as soil. When we use that fertilizer, when we apply that fertilizer in the soil, it will supply these heavy metals also to the soil. Then sewage sludge, metal mining and smelting, pesticide application, then electroplating industry. Electroplating industry is a major source of this heavy metal discharge when toxic substances, toxic heavy metals like chromium, cadmium, etc. are discharged from electroplating industry. Then, of course, fossil fuel burning. Fossil fuel is also obtained from nature only. Fossil fuels contain all these heavy metals, whether it is coal or petroleum. They will contain heavy metals like mercury and cadmium. And we know that it's not easy to remove these heavy metals. Heavy metals cannot be degraded by any biological or physical processes, and they are persistent in the soil for a very long period. Okay, earlier we talked about that persistent organic pollutants. No? Just like that, these heavy metals also will remain in the soil for a very long time. So it will pose a long-term threat to the environment. We have two types of heavy metals, essential as well as non-essential. These essential heavy metals, they support the growth of plants. During the plant life cycle, these heavy metals like copper, iron, manganese, nickel, zinc, etc., they are required for physiological and biochemical processes. But if they are present in excess, they may become toxic. Then we have non-essential metals, heavy metals like lead, cadmium, arsenic, and mercury. They are highly toxic. They have no function in plants, so they are non-essential heavy metals. And they may cause environmental pollution and severely affect a variety of physiological and biochemical processes in crop plants. And also they can enter into the food chain through crops and accumulate in human body. We call this biomagnification. So as the heavy metals move up in the food chain, their concentration increases. That is why it is called biomagnification. So biomagnification, so it is posing a great threat to human health also. So because of, so it is a real problem. There is a need to take up remediation measures to prevent heavy metals from entering into the different components of the environment and mitigate the contaminated land. So here phytoremediation comes to our rescue. Phytoremediation can be effectively applied here also. So plants have the ability to absorb ionic compounds in the soil, even at low concentration through their root system. So you know that the plants extend their root system into the soil matrix and establish the rhizosphere atmosphere. The rhizosphere ecosystem is associated with the root zone, okay, to accumulate heavy metals and modulate their bioavailability. And in that process, that polluted soil can be reclaimed and soil fertility will get stabilized. These are the advantages of using phytoremediation. Some of the advantages we discussed in the beginning also. It is repeated here also. Economically feasible, it is environment and eco-friendly, applicability, prevents erosion and metal leaching. It also improves soil fertility. These are the advantages. Then just like the organic chemicals, here also, the first step is anyway, uptake and translocation of heavy metals. So as in the case of this chlorinated organics, a series of processes are involved in the accumulation of heavy metal in plants, including heavy metal mobilization, root uptake, xylem loading, root to shoot transport. Anyway, root to shoot means from root to the leaf, root to shoot transport, then cellular compartmentation, sequestration, etc. And these heavy metals, mostly they exist as in the insoluble form. They are not easily soluble in water, which is so these heavy metals, most of them will not be available to the plants. So the bioavailability is less. But the plants can increase their bioavailability by releasing a variety of root exudates, some kind of enzymes and other substances excreted by the root. So they can increase the bioavailability. So which can change the rhizosphere pH and it can increase heavy metal solubility. So the bioavailable metal is sorbed at the root surface. So first it is the heavy metal is made bioavailable. So then bioavailable metal is sorbed to the adsorbed to the root surface and move across the cellular membrane into the root cells. Okay. And the uptake of heavy metals into roots occurs through two pathways. Two mechanisms are there. One is called the apoplastic pathway, which is a passive diffusion process. And the second one is a symplastic pathway, which is a active transport pathway. So there is a passive diffusion and active transport pathway. So the common uptake of heavy metals via symplastic pathway, it is an energy dependent process and is mediated by metal ion carriers or complexing agents. So then once they enter the root cells, heavy metal ions will form complexes with various chelating agents like organic acids. Several organic acids are present in the plants. So this they're called chelators. And they form a complex with these chelating agents. And such complexes, including carbonate, sulfate, and phosphate precipitate, are immobilized in the extracellular space. That is extracellular space. They're called the apoplastic cellular walls, or in the intracellular spaces. There, these complexes will get immobilized. Then the metal ions sequestered inside the vacuoles. Vacuoles are intracellular spaces. So the metal ions sequestered, stored inside the vacuole, may transport into the steel and enter into the xylem stream via the root symplasm, and subsequently translocated to the shoots through the xylem vessels. So they travel through the xylem vessels to the shoots, and they are transported and distributed in leaves where the ions are sequestered in extracellular compartments or plant vacuole. Then these heavy metal ion transporters. So a number of this uptake and translocation of heavy metals in plants is mediated by a variety of molecules, including metal ion transporters and complexing agents. So these specialized transporters are nothing but the proteins. They are known as channel proteins or hydrogen-carbon coupled carrier proteins. They are located in the plasma membrane of the root cell and are essential for the uptake of heavy metal ions from soil. And they can transport specific metals across cellular membranes and mediate efflux of metal translocation from roots to shoots. Anyway, I will just rush through. I think we have to stop at 3:30, no? Yeah, so I will rush through the presentation. So this is more about this heavy metal ion transporter, specialized transporters. Then, as in the case of organic substances, heavy metal detoxification, it is a key prerequisite for the implementation of phytoremediation. And plants have two defense strategies to cope up with the toxicity of heavy metals. One is called avoidance. Avoidance is a defensive strategy, and the other one is called tolerance. So by these two mechanisms, avoidance and tolerance, plants manage to maintain the cellular concentration of heavy metals below the toxicity threshold. If it goes beyond a particular level, these heavy metals will become toxic. So avoidance. Avoidance means it refers to the ability of plants to limit the uptake of heavy metals and restrict their movement into plant tissues through root cells. That is called avoidance. It is a first line of defense at the extracellular level through different mechanisms like root sorption, metal ion precipitation, as well as metal exclusion. Then you have tolerance. Once the heavy metal gets entry into the cell, cytosol, it is cytoplasm in the cell, tolerance strategy is adopted by the plants to cope with the toxicity of accumulated. It is the second line. So first line of defense is at the extracellular level, whereas tolerance, which is a second line of defense, is at the intracellular level through various mechanisms like inactivation, chelation, and compartmentalization of heavy metal ions. So when excess heavy metal ions are accumulated inside the cell material, the cytosol, plants have to detoxify in order to minimize their toxicity effects. So this is mainly achieved through one mechanism, that is chelation by complexation of heavy metal ions with the so-called ligands. There are certain compounds, metallic compounds. So they form a complex compound, complexation of heavy metal ions, the ligands, and through chelation, so chelation, the concentration of free metal ions are reduced to relatively low levels. So these are the organic compounds, ligands involved in heavy metal chelation, organic acids, amino acids, etc. Then after chelation, the complexes of ligands with the heavy metals are actively transported from the cytosol into inactive compartments like the vacuole, where the complexes are stored without toxicity. So here you can see, this is a schematic diagram showing the uptake, translocation, and sequestration of heavy metals in plants. These are the detoxification mechanism. Here are all, these are all part of the cell. Vacuole is a component of the cell where this that detoxified material is, that complex material is stored, then the xylem transport, and finally it will reach the leaves through the xylem. Then there are several phytoremediation strategies for heavy metals. Some of these terms were discussed in the case of organic chemicals also. We have phytostabilization, phytoextraction, phytovolatilization, phytofiltration. 4 strategies are there. But among these, phytoextraction is the most suitable for plants. The strategy which the plants use to extract and remove heavy metals from soil. Other methods are also efficient, but this is more efficient than the other strategies. Phytoextraction, phytostabilization. Anyway, we will focus more on phytoextraction. So phytoextraction means it's the use of plants to take up contaminants from soil or water, then translocate and accumulate these contaminants in their above ground biomass. It will not be in the root. So these contaminants will be translocated and accumulated in the above ground biomass. It may be in the stem, it may be on the leaves, etc. And the phytoextraction is considered as the most important phytoremediation technique for the reclamation of heavy metals and metalloids from the polluted soil. Several recent studies have showed the efficiency of this phytoextraction method. Whereas it is anyway different from phytostabilization. In phytostabilization, plants only temporarily contain heavy metals. They below ground. They remain below ground. So phytoextraction is in fact a permanent solution for the removal of heavy metals from polluted soil. It involves a few steps like mobilization of heavy metals in rhizosphere in the root zone, uptake of heavy metal by plant roots, then translocation from roots to aerial parts of plant, sequestration and compartment of heavy metal ions in plant tissues. These are the steps involved. And again, the efficiency of phytoextraction depends on plant selection, plant performance, heavy metal bioavailability, soil, and rhizosphere properties. Appropriate selection of plant species is very important. These are the characteristics of plant species suitable for phytoextraction: high tolerance to the toxic effects of heavy metals, high extraction ability, they should be fast growing, they should have abundant shoots and extensive root system, good adaptation to prevailing environment, they should be highly resistant to pathogens and pests. These are the characteristics of plants plant species which can be used for phytoextraction. So among these characteristics, the metal accumulating capacity and the above ground biomass are the key factors. So we have even certain hyperaccumulator plants which can accumulate heavy metals in above ground parts to a greater extent. Some such plant species are also there. Here also this list, here you can find the names of a few plant species used for metal accumulation. Then of course phytovolatilization from the transpiration process. Then phytofiltration is also there. Phytofiltration is not applicable for soil. It is for applicable for contaminated surface waters or wastewaters. It is called rhizofiltration, phytofiltration. Terrestrial plants, although sunflower. For phytofiltration, we can use all these with aquatic plants. Then also, there are attempts to improve the performance of plants which are used in phytoremediation. Already in the case of organic chemicals, we have discussed techniques like, we have discussed about transgenic plants, which are more efficient in decontaminating soils with organic chemicals. Here also, in the case of heavy metals also, we can think of such techniques to improve the plant performance. So here you can see the techniques. Here also you can see genetic engineering, transformation, then microbe-assisted phytoremediation. These are some recent developments which are employed to improve the phytoremediation efficiency of plants. Now, so we have discussed about the mechanism of phytoremediation with respect to organic chemicals removal and heavy metal removal from soil. But we have a lot of potential options are there, but the phytoremediation research is still in its infancy. A lot of work is yet to be done. Of course, we can do at field level also, so some attempts have been made, but the performance have not been that satisfactory. So we have to conduct more laboratory studies. Then we have to go for extensive research under field conditions. So the majority of the research has been conducted in laboratories under relatively controlled conditions for short periods of time. So we need extensive research under field conditions for long durations. And a limiting factor is we cannot have a single prescription. A specific phytoremediation prescription cannot be applied to every site with a certain chemical contaminant, because different site-specific conditions. We have to consider the conditions of the site, like the nature of the soil, climate, etc., which is suitable for the target plant. So there is no single prescription in phytoremediation. We have to carry out